Hello everyone, my name is Javier and today I'll introduce some ideas for using CRISPR to address climate change. We will focus especially in coral reefs. These beautiful ecosystems, despite covering less than 1% of the ocean floor, are home to at least a quarter of all marine life, including many fisheries that us humans depend on. The cornerstone of these environments are coral. These animals form calcium carbonate skeletons, which serve as habitats to the many other organisms in the ecosystem. However, recently, with the global rise of sea temperatures, these ecosystems are at the risk of vanishing. As we can see, at approximately higher than 29 degrees Celsius, a phenomenon called coral bleaching happens, and when these higher temperatures are sustained, it leads to the mass mortality of corals and eventually the loss of the whole ecosystem. So what is bleaching and how can we prevent it from happening? An important thing to understand is that corals depend on a symbiotic relationship with a dinoflagellate microalgae, also called zooxanthellae, which through photosynthesis provides the main source of carbon and other organic nutrients for the corals. But as you can see, as the red groups in the figure, under heat stress, the algae produce damaging reactive oxygen species, which force the corals to expel the symbionts. Recently, a revolutionary genetic engineering tool known as CRISPR-Cas was discovered as a bacterial immune system against viruses. It works with the interaction of two main components, a Cas protein capable of introducing a double-stranded break in the DNA, and a guide RNA complementary to a specific DNA sequence to which it guides the Cas protein for cleavage. So once we introduce a targeted double-stranded break, the cell can undergo two DNA repair mechanisms, non-homologous end joining, error-prone and introducing unpredictable indel mutations, often used for knocking out genes, or alternatively, if we supply a template DNA with homologous ends, we can introduce a specific sequence through homology-directed repair via the cell's homologous recombination. Moreover, advances in CRISPR technology have made possible the modification of the Cas protein, disabling the DNA cleavage domains and allowing for fusion onto other effector proteins of variance functions, for example, GFP or transcriptional regulators. Here, the dCas is directed upstream of the gene and fused to transcriptional activators or repressors, which regulate gene expression. Furthermore, just recently, we're able to introduce template DNA through homologous recombination with no need for that error-probable cleavage. So how could we use these revolutionary tools to prevent bleaching? Well, symbioses are a two-way street, just like our relationships, or at least I hope so. Hence why we would ideally target both corals and algae. Previous studies, as the one seen on the figure, have identified gene candidates from pathways such as photosynthesis, general metabolism, or growth that under heat stress are upregulated to a comparable intensity from baseline in red or downregulated comparably on intensity in blue. So taking in mind these gene candidates, we would ideally aim to enhance the symbiotic relationship on both organisms while diminishing the stress and damaging pathways that lead to bleaching. For instance, we would enhance the beneficial pathways by upregulating the beneficial gene expression with dCas activator fusion, or by introducing higher expression regulatory elements, or even enhanced genes through homology-directed repair. On the other hand, we could diminish the stress pathways through gene knockout, though if these are essential genes, we could instead downregulate their expression through dCas and repressors. The main challenge to these strategies is that we currently do not have a full understanding of coral bleaching, and very little genetic engineering has been done on both coral and zooxanthellae. Nevertheless, the solution to this could be CRISPR, as we could use it to uncover this complex mechanism through gene knockout to examine its effect on the organism, or by testing genetic components from other related organisms. Our final intention is to achieve higher bleaching thresholds so that corals and their symbionts are more resilient to heat or any other stressors. We expect higher thresholds with engineering coral or algae, but the highest when we combine both. Thank you very much for listening and feel free to ask any questions. Here are the references.